Hey Mustangs, in this video we're going to be taking a look at enzyme regulation, which really means we're going to be taking a look at how enzymes are controlled. How are they turned on? How are they turned off? How do we make sure that these enzymes that are involved are in our important metabolic chemical reactions, our catabolic, our anabolic reactions, how are they kept in check to make sure that these chemical reactions are occurring exactly as they should be occurring, to make sure that materials and energy are not wasted within the cell. So, there's two main ways to regulate your enzymes. The first is by turning the genes on or off that make the enzymes. So enzymes are a type of protein, and the instructions for building them are encoded in the DNA, encoded in the genes. Okay, so if you turn a gene on or off, so that means you turn transcription on or off for a specific enzyme, that is going to control the enzymes. And if they get turned on, then that means those enzymes are turned on and certain chemical reactions are going to occur within the cell. If you turn them off, that means certain chemical reactions are going to stop within the cell. So as an example here, and I like using this as a visual, and I use it many times in many videos here. So let's say you have an initial metabolite, an initial substrate that's going to be broken down, and you have, let's say, enzyme 1. So this is the first enzyme. That gets taken in by that enzyme. That gets broken apart and rearranged into something a little bit different here. Then enzyme 2 is going to grab that and pass it along. Usually there's a pathway. That's why we say anabolic and catabolic pathways that are involved in your metabolism. So usually more than one enzyme is involved in a pathway here. And then here, finally, here's enzyme 3. And so you get something like this. And then finally that passes on. And finally you get your final product that you're trying to get in this pathway, which might be something like that. Okay? So if you turn the genes on or off, turn transcription on or off for these enzymes, if you turn these off, everything stops. So you stop building these enzymes, which means this pathway stops, and these chemical reactions that are involved in this pathway will stop. The second way that enzymes are regulated is by simply turning the enzymes themselves on or off. So to do that, there are things called activators, and then there's inhibitors. So those are the two ways that you turn enzymes on or off. Now in both of these cases, 1 and 2 here, hormones can actually be involved. So hormones can actually cause a cell to turn transcription on or off for certain genes, which would then turn off a pathway. Now remember, when you involve hormones though, it's usually a temporary thing that's going on. You're temporarily changing what a cell is doing for a short period of time. And so in this case, you could turn the genes on or off for these enzymes, and that would stop or start a certain chemical pathway, metabolic pathway within a cell. Or sometimes turning the enzymes on or off could involve hormones as well, where you activate activators. That sounds weird. You turn on activators, or you activate inhibitors to actually turn an enzyme on or off. So let's take a look. We're going to focus here on these two right here. All right. So activators. Activators are what help an enzyme. So we say that they often turn an enzyme on. In the absence of activators, that means that the enzyme wouldn't work. The enzyme would not do the chemical reaction that it's supposed to do if there's a certain activator that's not present. So we call many activators cofactors. Okay, so cofactors usually bind to the enzyme, and then they help it catalyze the reaction. There's two main types of cofactors. There's inorganic cofactors, so usually minerals. Minerals are usually inorganic cofactors. So this is why in your food you need to make sure you get enough zinc, iron, copper. These are all activators. Certain enzymes could not do their jobs in your cells without these being present. And then you have what we call coenzymes. Coenzymes are often simply just organic, so inorganic versus organic cofactors, and that's usually our vitamins. So vitamin A, vitamin E, vitamin D, vitamin C, those are usually cofactors, activators, that if they're not present, then certain enzymes can't do their job. And again, this is why it's important to eat the right kinds of foods and make sure you're getting all of these things. Now in modern society, we often don't eat the right foods, and we don't get all the inorganic cofactors and the coenzymes, the vitamins that we need in our food. So we often take little pills that have these things instead because of our bad eating habits, you know. And if you did not do that, then you wouldn't have the cofactors necessary for certain enzymes to do their job. All right. So if we take a look here, here's an example of a cofactor. So here is the active site of an enzyme. So this big old part right here, like kind of a light blue color, is the enzyme. And then here's a coenzyme, cofactor. So this finally is the active site here. If you took these away, so you took this away, you wouldn't have the right shape for this enzyme to bind to its substrate. So the actual presence of these cofactors binding to this enzyme makes the active site become the correct shape in order for the substrate to bind here. Another example. So taking a look at here, so you might have a cofactor that once again binds and creates the correct active site for the substrates to be able to come along and actually bind, and the enzyme to therefore do its job. Then you have inhibitors. So to inhibit means to slow down or stop. So inhibitors slow down or stop enzymes from being able to do their job. Two main categories. You have competitive inhibitors. Here's the thing, to compete for. So think about what it means to compete. So competitive inhibitors. So if you take a look at this enzyme here, and here's in green the substrate, a competitive inhibitor binds to the active site. Competitive inhibitors basically block the active site. So if this substrate were to come along now and try to bind in here, it's not going to work. Induced fit will no longer happen. It's not the right shape for them to interact, so the enzyme can't interact with its substrate. So competitive inhibitors, the inhibitor actually competes for the active site and blocks the substrate from being able to get in there, and therefore the enzyme can't do its job. Then you have non-competitive, often referred to as allosteric inhibitors. So here, we see once again, there's our enzyme, there's the substrate, binds perfectly fine. With a non-competitive inhibitor, it binds to another site, not the active site. So it binds to another site on the enzyme. When it binds to this site, it changes the shape of the active site. So binding here at the allosteric site, not the active site, but the site away from the active site, causes the enzyme to change shape, and it no longer has the right shape in the active site. So if the substrate comes along, they don't go through induced fit, and the chemical reaction can occur. So you can stop an enzyme from catalyzing this specific chemical reaction. All right. Now, some inhibitors are irreversible, and we usually refer to those as toxins or poisons. So toxins or poisons are things that get in to enzymes and prevent them from being able to do their job. So if they're competitive, they permanently bind to the active site. If they're non-competitive, they permanently bind to that allosteric site and permanently change the shape of the enzyme. So sarin gas, pesticides, antibiotics, many of these things, that's what they do. They change the shape of enzymes and they stop a metabolic pathway. They stop certain chemical reactions. And the thing that makes these deadly is that these chemical reactions that they're stopping, that they're permanently stopping by turning off the enzymes, they're necessary for life. So if those chemical reactions can't occur, then the cells die, the organism dies. So an example, the sarin gas. So what the sarin gas does, it binds to certain enzymes that are involved in breaking apart neurotransmitters. So remember, neurotransmitters, you have the action potential that goes through the neuron. When the action potential gets to the end of a neuron here, we call that the axon terminal. It causes neurotransmitters to get released in the synapse. Those neurotransmitters float across and they bind to ligand receptors here and start the next action potential. Well, in order for this to reset basically and send another signal, there's enzymes that actually break apart these neurotransmitters. Those neurotransmitters get recycled and they get used again and again and again. Sarin gas binds to these enzymes. So these scissors represent the enzymes that are going to break apart the neurotransmitters to be recycled. The sarin gas binds to those enzymes and they can no longer break down and recycle, so you can't reset. So it basically shuts down your nervous system. So that's an example of a toxin or a poison right there. All right, feedback inhibition. We could also write for this one negative feedback. Okay, so feedback inhibition, many enzymes, many metabolic pathways that involve more than one enzyme. So this one involves 1, 2, 3, 4, 5 different enzymes that they take an initial substrate, break it apart, and keep passing along till you get your final end product. Now the way feedback inhibition works is this final end product here is going to actually go back and it's going to bind to an allosteric site on the first enzyme. So the final product made after passing from enzyme to enzyme to enzyme to enzyme, this final product, when enough of it builds up, it goes back, it binds to the first enzyme. And think about what an allosteric inhibitor does, or a non-competitive inhibitor does. When it binds to this site here, that enzyme is going to change shape and it's no longer going to bind, so it shuts down this whole pathway. So this end product feeds back. That's why we call it feedback inhibition. Feeds back and it changes the shape of that first enzyme, which then shuts down the whole pathway. All right, now our cells do not like to waste energy and materials. So enzymes are organized based on metabolic pathways, usually. So you'll find certain enzymes involved in certain anabolic and catabolic pathways in certain parts of the cell. So we call those enzyme complexes. So here we have an example, and they'll often be embedded in membranes. So you can see here's enzyme 1, 2, 3, and 4, and the substrate gets passed, changes it, pass to the next one, changes it, and keeps on going until you get your final end product. And if this was involved in feedback inhibition, this final end product would go turn off the first enzyme, and so on and so on. So in order to make sure that the cell doesn't waste energy, doesn't waste materials, enzymes are often organized into what we call complexes. Another really good example of this is here, what we see here, the electron transport chain. In the electron transport chain, the enzymes are involved and organized into complexes that can pass along products, in this case pass along electrons and pump out hydrogens here. So you have this very unique organization to make sure that the cell is run efficiently when it comes to enzymes.